Okay, hello guys. My name is Alamut Addasa. I am medicinal chemist in Addis Ababa University College of Health Sciences, School of Pharmacy. Today we will see the introduction for medicinal chemistry and some important terms that are used in medicinal chemistry. Uh, let's start by defining what medicinal chemistry is. Medicinal chemistry is the branch of pharmaceutical chemistry that deals with drug discovery, drug development, drug design, drug synthesis, preparation, and identification of new chemical compounds, new substances which are used for treatment, prevention, and the curing of human and animal diseases. But mainly, medicinal chemistry, it is defined as it is the science which deals with drug discovery and design of new and better therapeutic chemical and developing of these chemicals into new drugs. But also medicinal chemistry, it involves with the following very important things. The first one, medicinal chemistry concerned with intervention, discovery, design, identification, preparation of biologically active compounds. Medicinal chemistry also studies the metabolism of drugs, interpret the mode of action of drugs in the or at the molecular level. Medicinal chemistry also construct the structure activity relationship of drugs. We will see what is the meaning of structure activity relationship or SAR later. Medicinal chemistries, they make new compounds, they prepare new compounds, they determine the effect of drug in the body, biological process. Also, medicinal chemists, they can change or alter the structure of the compound for optimal, better effects and to minimize or to decrease the side or unwanted side effects of drugs. The side effect or unwanted side effects. Medicinal chemists also study the uptake, distribution, metabolism, and excretion of drugs. Let's see the areas of study. Medicinal chemistry, it involves, as I said earlier, synthesis of new compounds. This is one of the area of medicinal chemistry, synthesizing new important chemical substances, isolation from natural products or natural sources, designing the structure activity relationship for better therapeutic activity, for better desirable activity, to enhance the potency, to decrease the side effect. They can construct the structure activity relationship. Also, quantitative structure activity relationship. Absorption, transport, distribution, metabolism, and lot of things. This is studied by medicinal chemistry, illustrating their mode of action of drugs, interaction of drugs with the body, targets like receptors, enzymes, DNA. These are some of the areas of scopes of medicinal chemistry. Medicinal chemistry, it has the following convergence of disciplines. It interacts with the following disciplines, mainly in drug discovery and development: combinatory chemistry, synthetic chemistry for designing novel molecule, patent law, physiology, biochemistry for structure-activity relationship, physiology, pharmacology, immunology for pharmacodynamic studies. Also, physical chemistry, behavioral science, pharmacology, safety assessment, information technology. These are some of disciplines that are interacted with medicinal chemistry, mainly for drug design and discoveries. Mainly, most people, they agree that medicinal chemistry is a bridge between organic chemistry and pharmacology, because medicinal chemistry, it studies structure activity, the structure of drugs, and the pharmacology of drugs. Because of this one, also it is medicinal chemistry is the bridge between pharmacology and organic chemistry. Let's see some terms. New drug. What does it mean when we are saying that new drug? A new drug is a drug that has not been declared safe and effective by qualified experts under the condition prescribed, recommended, or suggested in a label, and that may be a new chemical agent. That is called a new drug. Another, hideout screening, or which is called in medicinal chemistry HTS. It is a highly automated robotic system that tests a small amount of large number of compounds against their potential targets. This is called HTS. It is robotic system that is used for drug discovery and development. And another, drug design, which is very important in medicinal chemistry. What does it mean when we are saying that drug design? It is the approach of finding a new drug by designing or by design based of the knowledge of its biological target. Therefore, designing, making, producing a new chemical substance based on the target site, based on the site of action. You have to have the knowledge of the target site to produce a new compound. Therefore, to design, you have to know the target. Therefore, designing or producing, preparing, or finding a new drug based on the target is known as drug designing. Drug discovery is the process of finding a new candidate drug, a new candidate substance, discovering, finding, getting a new substance with pharmacological activity, with biological activity. This is known as drug discovery. Drug development means the process of bringing a new pharmacological active substance or pharmaceutically active substance to market once a lead compound has been identified by drug discovery. Therefore, bringing a new chemical compound. Generally, drug discovery is mainly useful to get a compound with better safety, better stability, low cost of production, better efficacy, less side effects. We can develop the lead compound. A lead compound is the initial compound, the starting material in drug discovery. That is called a lead compound. Therefore, enhancing the safety, increasing the potency, increasing the stability, decreasing the side effects, unwanted side effects, decreasing the toxicity. This is drug development. We will see the example of penicillin. For example, penicillin is discovered by Alexander Fleming in around 1929. The first natural penicillin, it has lot of disadvantages, limitations, like allergy, instability in acidic media, instability for beta-lactamase enzymes or penicillinase enzymes, and it has narrow spectra. These are some of the limitations of the natural penicillin. Therefore, to avoid these unwanted side effects or limitations of natural penicillin, medicinal chemists, they develop or they produce new compounds from natural penicillin by enhancing the spectrum, by decreasing the unwanted side effects like allergies. For example, amoxicillin. For example, we have large number of these penicillin derivatives, flucloxacin. Therefore, these are some of the examples of drugs that are discovered or developed by this process, increasing the safety. Therefore, drug development means increasing the desirable effects, decreasing the undesirable effects. This is called drug development. Another, there are different tests in medicinal chemistry and pharmacology, also toxicology. In vitro test, meaning that testing the substance in an artificial environment or test tube or culture media. This is called in vitro test. The second one is in vivo test. Here, testing the substance, your sample, in living body, in the living animals. Ex vivo, usually this indicates that doing the test on tissue taken from a living organism. These are some important terms. Drug delivery. What does it mean when we are saying that drug delivery? It is a method or process of giving, administering a pharmaceutical compound to achieve a therapeutic effect in human or animal. Giving, administering a new substance is called drug delivery. And structure-activity relationship. What does it mean when we are saying that structure-activity relationship? This is the major point that can differentiate pharmacology from medicinal chemistry. In medicinal chemistry, we will mainly focus on the structure of drugs. Therefore, a structure-activity relationship is the relation between the chemical structure of a drug and the pharmacological activity. Therefore, this medicinal chemistry, it will studies about structure-activity relationship and the pharmacological activity of that drug. Or in other words, structure-activity relationship is the change in structure will changes the pharmacological activity of drugs or biological activity of drug. When you change the structure of a given drug, there is most of the time there is a change in activity. This change in activity is mainly studied by structure-activity relationship. We will see this one in detail later. And some other definitions. What are drugs? What are drugs? Drugs are compounds, substances that interact with a biological system to produce a biological response. There are different definitions for drugs. There is no single precise definition, as there are different meanings in drug control law, in government, regulations also, in medicine, in colloquial usage, there are different definitions. But In pharmaceutical science, we define this drugs, or a drug is a chemical substance used in the treatment, cure, prevention, or diagnosis of disease, or used to otherwise enhance physical or mental well-beingness. This is the definition of medicinal chemistry. And classification, how drugs they are classified. Drugs can be classified according to various criteria. Among this one, the first one is drugs, they may be classified based upon their origin or their source. Maybe synthetic origin, natural origin, maybe semi-synthetic origin. The first classification. Drugs classified based upon their origin, based upon their source, from where they were derived from. Synthetic origin, for example, aspirin is synthetic one, paracetamol is synthetic one. Those drugs that are synthesized in the laboratory, they are called synthetic. Laboratory means in industries, pharmaceutical industries. These are called synthetic origin. By their origin, they may be natural. There are many drugs that are derived from natural sources. For example, plants. A lot of drugs nowadays, they are derived from plant origin. A lot of drugs like digoxin, quinine, also ephedrine, colchicine, they are plant origin. Those drugs that are derived from plants, they are natural. Animal origin, this is also natural one, which is from based upon their origin. Insulin is the animal product. Also gonadotropins, heparin, thyroid extracts, these are animal origin. They were derived from animals. The other natural origin is minerals. There are many drugs that are derived from minerals. Iron, calcium, magnesium, aluminum, sodium, potassium. Drugs that contain these elements, they are derived from minerals. The other natural origin, those that are derived from natural sources, microorganisms, those drugs that are derived from microorganism. For example, penicillin, tetracycline. These are some of the examples of drugs that are derived from natural source. Therefore, the first one is synthetic origin. The second one is natural origin. The third one is semi-synthetic origin. Based upon the origin, they are classified as natural, synthetic, and semi-synthetic drugs based upon their origin. Semi-synthetic drugs are drugs that are partially synthesized from natural one. For example, 6-amino-penicillinic acid, an example of these drugs. Therefore, the first classification of drugs is drugs, they may be classified based upon their origin, from where they were derived. The second one, drugs, they may be classified based upon their pharmacological action, like analgesic drugs, anti-pain, those drugs that relieve pain, bronchodilators, hypnotic drugs, antiviral drugs, anti-malarial drugs, anti-arrhythmic drugs, anti-anginal drugs, antibacterial, anti-allergic drugs. This classification is based upon the pharmacological action. What is the action of drugs? Pain treatment, killing bacteria or inhibiting the growth of bacteria, antibacterial, anti-malarial. This is the second classification. The first one is based upon their origin. The second one is based upon their pharmacological action. They may be also classified. The third one, drugs, they may be classified based upon their chemical structure, based upon their common structure, like steroids that contain, for example, those drugs that contain cyclopyrrhydrophenetrin ring, like estrogen, progestin, or progesterone. These are called steroids or steroidal drugs. They have common structure. Also, beta-lactams. They have a common structure, the lactam structure. Also, carboxylic acids, alcohols, phenols, glycosides. For example, we can see cardiac glycosides like cubane, covaltoxin, also digoxin, digitoxin. They are called cardiac glycosides that are used for treatment of CHF, congested heart failure. They have the same structure. They have similar structure, common structure. Also, some drugs that are classified based upon their chemical structure, quinolones. There are many examples under this, like fluoroquinolones that are antibacterial drugs like ciprofloxacin, gatifloxacin, inoxacin, norfloxacin, sparfloxacin. They have the same structure because we will see the structure their portion. Therefore, drugs that have the same structure, the same nucleus, the same parent structure, they are classified based on their chemical moieties, chemical structure. The fourth one, drugs, they may be, fourthly, may be classified based upon their site of action, target of action, where they will act. Drugs that will act on the adrenergic nervous system, they are called adrenergics, or they are called sympathomimetic drugs. Drugs that will act on the dopaminergic nervous system, they are called dopaminergics. Drugs that will act on the parasympathetic nervous system, they are called parasympathomimetics or cholinomimetics, cholinergics also. Therefore, based up on their sites of action, based up on their target, they may be classified. Therefore, generally, drugs, they may be classified based on their origin, maybe natural, synthetic, or semi-synthetic. The second one, drugs, they may be classified based up on their pharmacological action. The third one, drugs, they may be classified based upon their chemical structure. And drugs may be classified based upon their target of action. This is about how drugs, they are classified. Then let's see drug body interaction. The interaction between the drug and the body, they are conveniently classified into two major classes. The interaction between the drug and the body. This may be pharmacodynamic or pharmacodynamics or pharmacokinetics. Pharmacodynamics means what the drug does to the body, the effect of a drug, mode of action. This is called pharmacodynamics, the effect of a drug in the body. The second one, pharmacokinetics, what the body does to the drug, like absorption, distribution, metabolism, elimination. This is called pharmacokinetics. These are some important things that body and drug interactions. Some important things. Therefore, also there are some factors that will affect the drugs to reach their active site. Absorption, distribution, metabolism, and elimination. You can see this one mainly in pharmacology. Their definition also for beta. You can see also here the definitions we have. These are the major factors that will affect the ability of a drug to reach their site of action. And there is also the interaction between drug and receptors. This may be agonistic interaction or antagonistic interaction. Let's see these two important terms in pharmacology, also in medicinal chemistry. What are agonists? Agonists are drugs that activate a receptor, that potentiate receptor. They are called agonists. They may be partial agonists or full agonists. Partial agonists are agonists that cannot elicit the maximal or maximum response, that cannot produce the maximum response. They are called partial agonists. Full agonists are agonists that can produce maximum response, maximum efficacy. They are called full agonists. Maybe also based upon their action, they may be direct acting or indirect acting agonists. Direct acting agonists, they will directly act on the receptor. But indirect agonists, they will stimulate another receptor and they will potentiate the receptor, the desired receptor. Therefore, those that will directly act on the receptor and indirectly potentiate the receptor. These are direct and indirect agonists. And antagonists. These are drugs that does not or do not activate the receptor. They block the receptor. This may be irreversible or reversible antagonists, or maybe competitive antagonists or non-competitive antagonists, which will compete with the agonist for binding. These are also some of drugs. The interaction between the drug and the receptor may be classified as agonist and antagonists. And dose response curve, that is very important in pharmacology. From the dose response curve, we can determine the efficacy, potency of drugs. What is efficacy? Efficacy is nothing but is the response that is generated. If you have the dose response curve, the dose is on the x-axis, the response is on the y-axis. Those drugs that produce maximum response, maximum efficacy, that means greater y-axis, they are called efficacious drugs. Drugs that produce maximum response, they are called efficacious. Efficacy is the maximum effect that a drug can produce regardless of a dose. It doesn't depend upon the dose of a drug. Therefore, the maximum response is called efficacy on a dose response curve. And Potency is the amount of a drug that is needed to produce a given effect. The lower the dose needed to produce a response, the more potent is a drug. Therefore, the lower the dose, the higher the potency. Drugs that can produce efficacy, drugs that can produce response at low dose, they are called potent drugs. Let's see some examples. Here, we can see here. This is the difference between potency and efficacy. Here, in these curves, there is the y-axis. This is the y-axis. This is the x-axis. Then, in this y-axis, we can measure the response. Those drugs that produce maximum response, they are called efficacious drugs. For example, I have A and B. This is A. This is B. If you see the level of B, it is greater than A. Those drugs that can produce maximum response, they are called efficacious. In another word, B is more efficacious than A. And the dose, to determine potent drugs, you have to see the x-axis. Those drugs that produce response at low dose, they are potent. Here, this is A, this is B. Therefore, from A and B, A produces effect, response, but the dose of A is less than B. Therefore, A is more potent drug than B, but B is more efficacious than A. Therefore, this is used to measure the efficacy. Here, this is the efficacy difference, the y-axis between A and B. The x-axis, the difference between A and B, to measure the potency. This is how to identify drugs which are efficacious and potent. Also, I have here these examples, A and B. Here also, A and B. These are some examples for you. Therefore, here, this mainly, let's do the fourth one. Here, I have A and B. I have A and B. This is A, this is B. When you see the two drugs, A has more y-axis than B. Therefore, A has greater response. Therefore, A is more efficacious than B. Also, let's see the potency of these two drugs. When you see the potency of A and B, you can measure on the x-axis to determine the potent drug. Here, A is around 0 point something. B is around 10. Therefore, those drugs that produce response at low dose, at low concentration, they are called more potent drugs. Therefore, this is some of the things that are used for identifying more potent and more efficacious drugs. I have also some exercise for you. Therefore, I have curve A and curve B. Compare the potency and efficacy of the following hypothetical dose response curves. I have the first curve A, I have X, Y, Z. Therefore, when you see the potency, X is more potent than Y. Y is more potent than Z. Or X is greater than Y, Y is greater than Z in decreasing order of potency. In increasing order, the potency, Z is less than Y, Y is less than X for curve A, the potency. When you see the efficacy, it's almost, when you see X and Y, it's like similar. You can measure for more. Therefore, here X and Y, I think it is almost similar. Therefore, if the level of x and y in this curve, or if their y-axis are similar, we can say that x and y have equal efficacies. And x and z has similar efficacies. When you see x and z, they have almost similar efficacies. You can measure for more. Therefore, x has greater efficacies than y. Also, z has greater efficacies than y. But when I saw the curve, x and z, they are almost equivalent. Therefore, you can go. Then, curve B. This curve. Curve B means this curve. Then, this curve, A, B, C, D, they have equal y-axis. This means that they are equally efficacious. They have the same response, almost similar response. Those drugs that have similar response, they are called with the same efficacies. When you see A, B, C, D, their dose is different. Those drugs that produce efficacy or response at low dose, they are called potent drugs. Therefore, A is more potent than B, B is more potent than C, C is more potent than D. In decreasing order, A is greater than B, that means potency. B is greater than C, C is greater than D. In increasing order, D is less than C, C is less than B, B is less than A. This is the order to identify these drugs. Also, therapeutic index. What is therapeutic index? Or sometimes it is called chemotherapeutic index, TI. It describes a relation between the dose of a drug that can cause lethal or toxic effect with the dose that can cause therapeutic effect. It is also called therapeutic ratio. It is the ratio between LD50 and ED50. What is LD50? It is the minimum amount of a drug that can cause adverse effects in 50% of population. ED50 is the quantity or the amount of a drug that can produce desirable therapeutic effect in 50% of population. Therefore, this is a relation between the two. And the other that is very important in medicinal chemistry, the nomenclature. Nomenclature means naming of drugs. How drugs they are named. Nomenclature. Generally, drugs they have most of the time people they have or they agree that there are three or more names. Here I will show you that there are 4 names for drugs, or there are 4 nomenclating ways for drugs. The first one is called code number or code designate. What does it mean? Code number or code designate. This code number is formed with the initial name of a laboratory, the chemist or research team that prepare or test the drug at the first time in the laboratory, followed by number to represent a drug. This is the name of a drug that is initially given in the laboratory the first time to identify your sample from other samples, to identify your drug new compound from other compounds. You can give letters or names or symbols. This name is called code number or code designate. It does not identify the chemical nature or the structure of a drug. By the way, one of the disadvantages of code number or code designate is it doesn't imply, it doesn't indicate the chemical structure. It doesn't indicate the chemical behavior of the compound. Also, structure of the compound. And it is discontinued as soon as an adequate name is given. That is the laboratory name. Mainly people, they use their first letter, mainly their names, first letter, and their father name, or their country code. For example, fluconazole is the most popular antifungal drug. It is code number or code designate is indicated here, UK 47 something. Therefore, this is the code number. This naming system or these names, they can be removed as soon as the adequate name or correct name is given. This name is called the laboratory name. The second one, chemical name, that is common. You can get these chemical names of different compounds, different drugs in books and Google. But this is not used. What is chemical name? This chemical name of a drug is given by the rule that is called IUPAC scientific naming system based on the molecular structure of the drug. They describe the substance chemically. They indicate the chemical structure, molecular structure of a drug in terms of atoms, molecules, and accompanied by diagram of the chemical structure of a drug. They indicate the chemical property, the chemical structure of the drugs. They are useful, very useful for chemists or technical peoples because they provide the precise arrangement of atoms and atomic groups in the molecule. But they are long and difficult and not suitable for common use, mainly in prescription, because they are very long. There are some drugs that contain more than 30 letters. Therefore, it's very difficult to prescribe, mainly in common use, very difficult. Therefore, this is not used. Chemical name is not mainly used in drug naming system, but is one of the name that is used. But you can get this name is chemical names in books, but in prescription, in common usage, we cannot use this chemical name. The reason is that because they are very long to use. They are very long for prescription. We can use another naming system. That is the third and the fourth one. Then, for example, for chemical names, I have two examples. For paracetamol, the chemical name paracetamol. The chemical name for paracetamol. These are some examples. There are some drugs that contain more than 30 letters. This is some examples. For paracetamol, the chemical name is N4-hydroxyphenyl acetamide. N4. At the fourth position, there is hydroxyl group. Therefore, it indicates the number of atoms, the type of atoms, the chemical structure of the compounds, chemical naming system. It tells the atoms that are found in that molecule, in that drug. Also, in the case of aspirin, 2-acetoxy-benzoic acid. Benzoic acid, 6-membered cyclic compound that contain carboxylic group at one position and ester group, acetoxy group at 2 position. This indicates that the structure of a compound. Simply, you can get the chemical property of that compound by using the chemical nomenclature, the chemical name. But it is not suitable for prescription, not suitable for common usage. This is the limitations of the chemical naming. The third one is called the proprietary name. Property means belongingness of something. It is also called trade name, brand name, serial name, trademark. These are the names that are given for brand names. In common usage, we can use simply brand name. That means trade name, proprietary name. These are names given to the drug that is discovered or developed by pharmaceutical companies and are their property or trademarks of the manufacturer company. Therefore, the brand names are given by manufacturer, unlike that of generic names. They are names that are given by individual manufacturer. In fact, a drug may have more than hundreds of brand names, but it has a single generic name, because different manufacturers, they can use their brand names. These names are patented or copyrighted by terms selected by a manufacturer to designate particular product. Once a new compound or a new drug, usually known as period drug, is discovered, the company files for a patent. It should be, this brand name always should be written with capitalization of the first letter of each word of the name, always. Not only when it comes or appears before sentences. Always the brand names, they are written in capital letter. In front of the sentences, in the middle of the sentences, always the brand names are capital. Also, there is the brand copyright, that is R, at the superscript to indicate this name is the brand name. Most of them also they are bold and drugs. Therefore, this proprietary name or trade name indicates that the name of the manufacturer. That is to identify the product. For example, Panadol, Tylenol, these are the brand names for paracetamol. A compound or a drug may have more than 100 names, brand names, because different manufacturers, they can produce this compound by using different their trade names, their brand names. Mebendazole, it is the generic name for anti-helminthic compound. It is also marketed as Vermox and Qmox. These are brand names. This is manufacturer name, but the common name is, or generic official name is, Mebendazole. Therefore, this is some of the differences between the drug nomenclature. The fourth one, which is called generic name, common name. Sometimes it's also called official name, non-proprietary name, which is not belongs to a given manufacturer. Common name, official name, generic name. These are the same. These refer to the commonly established name which a drug is known as an isolated substance, irrespective of a manufacturer. It should be simple, concise. The first letter is not capitalized like that of brand names. There is no mainly R at the superscript. It only capitalized when it appears at the beginning of the sentences. It is chosen by official agencies such as US adopted names or councils, American medical associations, American pharmaceutical associations, pharmacopoeia, also US Food and Drug Administration, WHO. These are responsible organization for giving official names for drugs. Therefore, these are the 4 names for drugs. Drugs, they may have 3 or more than 3 names. This includes trade name, code name or code designate name. The second one, chemical name. The third one, trade name, commonly called brand name or proprietary name, serial name also. This fourth one is common name, official name, or generic names that are used. But the most common are the brand names and generic names that are the most common. They are also concise and simple to use for prescription, unlike that of chemical names. And genesis of drugs. We say that medicinal chemistry is a discipline that is mainly involved in drug discovery and development. Then, how drugs they are discovered? What are the methods, approaches that are used to discover new drug? How can one discover? What are the methods that are used in drug discovery? Therefore, I define that drug discovery means finding, getting a new compound, a new substance. Whatever. This is called discovery. Therefore, how one can discover a new drug? The first one is drugs, they can be discovered by serendipity. Serendipity means accidental discovery, accidental finding of a new drug, a new substance with non-biological activity or with biological activity in any place, maybe in hospital, in research areas, in pharmacies, wherever it is, in universities, in colleges, wherever. This is called serendipity. Accidental, by chance, getting a new compound by chance is called serendipity. For example, the discovery of penicillin by Alexander Fleming. He is doing during that time, his master thesis by culturing bacteria. That bacteria is contaminated with fungus. Then, during that time, before 1929, penicillin is not antibacterial, but is antifungal. When he applied this antifungal drug for those bacteria culture, all bacteria, they were dead. Then he and his professor, they understand that in addition to antifungal activity, this compound, it may have antibacterial activity. Then they discovered the antibacterial activity or antibiotic activity of penicillin. Such type of discoveries, they are called serendipity. There are many drugs that are discovered by this mechanism. For example, isogramine, which was the prototype for lidocaine discovery, local anesthetic drug. Also, anti-inflammatory drugs, analgesic drugs, antipyretic drugs, like phenylbutazoles, they are discovered by serendipity, meaning that serendipity means accidental determination, accidental discovery of a new compound in any place is called serendipity. Therefore, the first one is drugs, they can be discovered by serendipity. The second one, random screening. What does it mean? Random screening means collecting or submitting large number of substances or chemicals, maybe plant origin, animal origin, whatever it is, submitting or collecting large number of chemicals, substances, by hoping that some of these chemicals, they may show activity. It is hoping. Several compounds, they may be submitted. Then checking the activity by hoping that some of these compounds, some of these substances, some of these chemicals, they may show activity. It is not very rewarding, tiresome, difficult. For example, to discover, to check the anticonvulsant activity of some chemical substance, they submitted or they collect 500 chemical compounds to check whether these 500,000 chemicals, they may show anticonvulsant or antiepileptic activity. Very difficult. From these 500,000, there may be only one, maybe also zero. It's very difficult, time consuming, very difficult way of drug discovery. In fact, there are some drugs that are discovered by this mechanism. Therefore, this is not rewarding one. The second way of discovering a new drug is called random screening. Collection of large number of substances by hoping that some of them may show activity, pharmacological activity. Maybe anti-malarial, anti-leishmanian, antiviral, whatever it is. This is not rewarding. The second one. There are also, in fact, there are some drugs that are discovered by this mechanism. The third one, rational directed random screening. Rational directed random screening. This is the third one. It is better than simple random screening. Do you know that? Rational directed random screening. Therefore, here, collecting or submitting chemical substances based up on the rationale behind some discoveries. For example, penicillin was discovered as antibiotic drug in 1929, around 1929. Therefore, by this mechanism, people's, scientists, researchers, they collect penicillin-like compounds that contain beta-lactam ring. Because penicillin showed antibiotic activity. Therefore, those penicillin-like compounds, those drugs, those substances that contain the beta-lactam ring, they may show antibiotic activity. They may show. Therefore, collecting drugs based upon the rational direction or rational directed method is another way of discovering a new drug. Therefore, they discovered more than 600 drugs, antibiotic drugs from penicillin by using rational directed drug discovery. Rational directed drug discovery. Because penicillin showed antibiotic activity, antibacterial activity. Therefore, drugs that are structurally similar with penicillin, they may have antibiotic activity. By hoping that, they collect many compounds. Among these compounds or substances, 600 of them, they showed activity. Some books also said 100, more than 1,000 of these compounds, they showed activity. Therefore, this is better than random screening. Random screening. Collecting drugs based upon the rationale behind the discovery of another drug. And natural products, using natural products, animal products, plant products, microorganisms, minerals, mainly those compounds, natural products that are greater affinity for their receptor. Using these natural products, we can discover a new compound. For example, morphine is a natural compound. For example, also atropine, ephedrine. These compounds, they have greater affinity for their receptor. They have greater binding affinities. Therefore, checking these compounds for other activity. By using natural products, we can discover, we can identify, or we can find a new compound, a new drug by using a natural source. The fifth one, using drug metabolites. Using drug metabolites. Some drugs, when they are metabolized, they produce inactive substances. Some drugs, they produce active substances. Therefore, using drug metabolite as a lead compound, we can discover a new drug. A lead compound is the starting compound, the initial compound for drug discovery. Therefore, for example, paracetamol is discovered by lead compound metabolism. Therefore, here acetanilide, when it is metabolized, it produces acetaminophen, paracetamol. Acetopentadine, when it undergoes metabolism, it produces paracetamol. Therefore, using drug metabolites, we can generate, we can find a new compound, a new substance. This is also another way of discovering a new drug. The sixth one, to discover a new drug, clinical observation of drugs, mainly the side effects of drugs. Some drugs, they may have additional effects. Observing these additional effects, we can discover a new drug. Innumerable examples can be found in the literature, such as the hypoglycemic effect of some antibacterial sulfonamides. There are many examples. For example, these sulfonamides, antibacterial drugs, when they are structured, when they undergo structure activity modification, they produce sulfonylureas because they show some other activity in addition to antibacterial activity. Therefore, seeing the side effects of drugs, we can discover a new compound. Also, the last one, how to discover a new drug. Rational approaches. Rational approaches. This is the most common nowadays for drug discoveries. The key information that permits rational approach to drug design is the knowledge of the etiology of the given disease, or at least the biochemical process. Therefore, designing a drug based upon its binding site is called rational approach. There are many drugs that are discovered by rational approaches. Therefore, these are some of the methods that are used to discover new drugs. Also, to discover new drugs, there are also other methods. Molecular modification of existing drugs, or molecular manipulation, by modifying the structure of the existing drug. Modifying the structure. By changing the structure, we can discover a new drug. Here, the objective of molecular modification in drug discovery is, or lead structure modification is, to obtain drugs having more desirable property than the lead compound, the prototype. To increase potency, to decrease toxicity, to increase specificity, to increase duration of action, to increase ease of application, to increase stability, to decrease cost of production we can modify the structure. We can develop a drug. We can manipulate a drug, lead structure. Therefore, the objective of lead structure modification is to increase the desirable effects, to decrease undesirable effects. This is the major objective. Under this molecular modification and or molecular manipulation, we have two methods, two basic approaches: general process and special processes. Therefore, to discover a new drug, here you have to follow either the general process or special process. This is mainly if you have the lead compound in your hand. If you have the lead compound with the known pharmacological or biological activity, you can undergo drug development or modification. This can be done by either general or special processes. Under general process, there are two general processes that are mainly used in drug discovery. Therefore, to discover a new compound, we can use general process. For example, disjunction or molecular simplification. What does it mean? Disjunction or molecular simplification means, it means synthesis of a smaller and a smaller drug from bulkier ones. Systematic production or evaluation of bulky compound, changing the compound with the bulky structure into a smaller one. Here the objective is to increase potency, efficacy, to decrease side effects, or to increase the desirable pharmacological activity. During that time also you can get a new compound. By using disjunction or molecular simplification, we can discover a new drug. What does it mean? For example, look here. The first structure is cocaine. This is the structure of cocaine. We will see in chapter 4. Cocaine is previously the most popular type of local anesthetic drug, but later because of its addiction, it is not used further. This cocaine, it has three major side effects. One, it is highly addictive drug to use as a local anesthetic drug. The second one, it is unstable compound. Its duration of action is very short. The third one, it is highly allergic drug. Therefore, to overcome these three unwanted side effects of cocaine, it is better to modify the structure, because on the structure of cocaine, there are side effects that are responsible for side chains that are responsible for the side effects of cocaine. By removing these side effects, they produced a better safe compounds. Here, look, this is procaine. This is cocaine. There are many steps that are used in the production of procaine from cocaine. Here, there are many steps, not only one or two steps. Several steps are were used. Therefore, here, the objective is to decrease the side effects and undesirable effects of cocaine. Here, when they saw the structure of cocaine, it contains the carbomethoxy group. This is called the carbomethoxy group. It also contains the tropine moiety. This is called the tropine moiety. And there is a benzoyl, the third one, the benzoyl group. They saw the three moieties to modify this structure. Therefore, when they remove some groups, mainly when they remove the carbomethoxy group, the drug is free from addictive liability. Then they say that what is the cause for cocaine addictive liability? Then they check that the presence of the carbomethoxy group is responsible for cocaine addictive liability. Therefore, it must be removed. And they generate, there are many steps as I told for you. They generate this compound. Here there is no carbomethoxy group on procaine. And I said for you that this compound is allergic. Same thing. Why it is allergic? Because this compound is allergic because there is a tropine moiety. Therefore, if we remove the tropine moiety, the allergic problem may be minimized. Therefore, they also remove this tropine moiety to produce this compound. Therefore, here, disjunction or molecular simplification is production or synthesis of smaller and smaller drugs from the bulkier one to minimize the side effect to enhance potency or to enhance potency. Therefore, this compound is free from addiction because it lacks the carbomethoxy group. It is to some extent free from allergy because it lacks the tropine moiety. Why this cocaine is unstable? Because there is a ester group. The same thing, this derivative procaine, it has also a ester group. Because of this one, it's unstable. One of the major problems of procaine is unstable and less potency than cocaine. Therefore, to discover a new drug by molecular modification or manipulation, we can use this method also, disjunction or molecular simplification, to bring better, safer compound. The second one, conjunction or molecular association. This is nothing, combination of two or more than two drugs by using either hydrogen bond, ionic bond, covalent bond. There are many drugs that are formed by this mechanism, many combinations, association or combination, molecular association. For example, cortam, the combination between lumefantrine and artemether, augmentin, that is discovered by molecular association between amoxyl or amoxicillin and clavulanic acid. They're just trying to give you examples of drugs that are discovered by this mechanism. There are many compounds, many drugs, conjunction or molecular association, SNP, many drugs we have. Therefore, what does it mean? How one can discover a new drug by conjunction or molecular association? Therefore, here, synthesizing or combining or associating different drugs by either covalent bond, ionic bond, whatever it is, to produce bulky substance. During that time, there may be change in activity. There may be change in efficacy of that compound. There may be change in stability of the compound. Therefore, here the objective is to increase efficacy or to increase potency or to increase duration of action. Therefore, here conjunction means combination of two or more than two drugs by these forces. This is classified into three major classes: molecular addition, molecular replication, molecular hybridization. There are three methods that are used in molecular association or conjunctions. Let's see each of them. What is molecular addition? Molecular addition means association of different moieties, different drugs, by using forces like ionic bonds or hydrogen bond. Ionic, which is called electrostatic force, or ionic bond or hydrogen bonds. This is called molecular addition, combining two or more than two drugs by using ionic or hydrogen bonding. Example here I have MedChem I Intro, ionic bonding, combining two or more than two drugs by ionic bond or hydrogen bonding. Different moieties, different drugs. The second one, molecular association or conjunction, molecular replication. Here, this is the method of discovering a new drug by combining identical drugs. Drugs that have similar structure. Therefore, combining identical twin drugs. Twin drugs. For example, the combination between salicylic acid. Two molecules of salicylic acid produces salicyl salicylate, dicumarol. Therefore, combining two identical twin drugs by using covalent bond, but not ionic bond, but not hydrogen bond. This is called molecular replication. Combining or associating two or more than two drugs by using covalent force. Identical, two identical, always identical drugs by covalent force. This is known as molecular replication. Molecular replication. Similar, identical twin drugs. The third one of conjunction or molecular association to discover a new drug is molecular hybridization. In this case, it is the process or the synthesis or association of two or more than two mixed drugs, different drugs, by covalent bond formation, non-identical drugs. For example, acetaminophenol, the combination between aspirin and paracetamol, different drugs by covalent force, not identical, non-identical drugs. Such type of discovery is called molecular hybridization. Then, what's the difference between molecular hybridization and molecular replication? What's the difference between molecular hybridization and molecular addition? What's the difference between molecular addition and molecular replication? Mainly the type of the drugs that combine or the bond that is used to combine or associate this. Therefore, in molecular hybridization, we can use different drugs by covalent bond. And molecular replication, we can use identical drugs with covalent bond. Covalent bond, identical replication. Sorry. Then, molecular addition with ionic bond, different. These are some of the processes that are used. Thank you for today. I'll continue the next portion, that is drug discovery, mainly molecular modification, special process. I'll come back.